In this video, I'm going to cover incomplete dominance, which is a type of inheritance pattern for some traits. So what incomplete dominance means, the prefix in means not. So it's we still have like a dominant and recessive allele. However, the dominant allele is not completely dominant. That's what incomplete dominance means. So when we have a heterozygous phenotype, that's where we're going to see a different phenotype come out. So we're actually going to have 3 phenotypes instead of just the 2, because when the dominant and the recessive allele are inherited together, the phenotype is going to be a blending of the 2 traits instead of the dominant taking over. So for instance, right, like Spoon has a baby with a fork. If the baby's heterozygous, it would be a blending of the 2, which is why it would be a spork. Some actual examples of traits that show incomplete dominance: in snapdragon flowers, you can have plants that have red flowers. You can have plants that have white flowers. And the heterozygous phenotype would be a blending of the 2, so that's where you see the pink flowers. In humans, we can have curly hair, we can have straight hair, and the incomplete or the heterozygous phenotype is going to be a mixture of the 2 or a blending of the 2, which is how you get the wavy hair. In dogs, right, we can have big dogs, we can have little dogs, and the heterozygous phenotype is going to be a blending of the 2, which is going to be a medium dog. In bunnies, we can have black bunnies, we can have white bunnies, and the heterozygous phenotype is going to be a blending of the 2, which is where you get the gray bunny. In dogs, again, we can have dogs with lots of spots, we can have dogs with little to no spots, and the heterozygous phenotype is going to be a dog that has some spots. It's the blending of the 2. So when we do an incomplete dominance problem, we are going to use different letters to represent each of the different alleles. So it's going to look a little bit different in the first couple steps compared to monohybrid, but it's done the same way. So for our first example problem, in morning glory plants, red flower and white flower alleles are incompletely dominant, with the heterozygous having pink flowers. The red-flowered morning glory is crossed with a white-flowered morning glory. What are the genotypes and phenotypes and their percentages of the offspring? So for our first step, right, we usually define our letters. But for incomplete dominance, I'm going to have you actually define all of your genotypes. So since I told you that red flower is going to be represented with an R, the only way to have red flowers would be to have 2 alleles that are the letter R. So RR is red flowers. White flowers, I told you, excuse me, to use the W allele. So in order to have white flowers, we're going to have WW as our alleles. And to be heterozygous, you would be RW, and that would be how you get the pink flowers. So that's my step 1. Instead of just telling me what R means and what W means, I want you to tell me what RR means, what WW means, and what RW means. We are crossing a red-flowered morning glory with a white-flowered morning glory, so we're going to have RR and WW as our parent genotypes. We can plug those into a Punnett square, and we should get all of these as our offspring. So for our genotypes, we see that every single one of these is heterozygous. So we'll have a 100% chance of the baby flowers being heterozygous. And for phenotypes, our W codes for pink flowers, so 100% of the baby plants will be or will have pink flowers. So what's nice about doing step 1 in this fashion is when you're going to do step 4, looking at the genotypes and the phenotypes, especially the phenotypes, these are defined for you in step 1. For the second problem, we have humans, straight hair, which is represented with an H, and curly hair, represented with a C, are incompletely dominant with the heterozygous having wavy hair. A straight-haired individual is crossed with a wavy-haired individual. What are the genotypes and phenotypes and their percentages for the F1 generation? Step 1, let's define our genotypes. So to have straight hair, I would have to be HH. To have wavy hair that's heterozygous, I would have to have HC, and then CC will code for curly hair. I have a straight-haired individual, so that's HH, crossed with a wavy-haired individual, which is HC. So these are our parental genotypes. If I plug those into the Punnett square, I would get these offspring. For my genotypes, because I can't really say homozygous dominant, homozygous recessive, because there really isn't a dominant and a recessive anymore, you can just say that 50% of the offspring will be HH and the other 50% will be heterozygous. And then for our phenotypes, HH codes for straight hair, so we'll have 50% with straight hair, and we'll have 50% with HC, which codes for wavy hair. If you want to try this problem on your own, hit pause and come back when you're done. Otherwise, I'm going to walk through it right now. So for the third example problem, in sheep, black wool, which is represented with a B, and white wool represented with a W, alleles are incompletely dominant, with the heterozygous having gray wool. Two gray sheep are crossed. What are the genotypes and phenotypes of the offspring? So let's start by defining our genotypes. To have black wool, you'd need BB. To have white wool, you'd need WW. And heterozygous would be BW, and that would code for gray. If I have two gray sheep being crossed, that'd be BW with BW. When you plug that into your Punnett square, these are the offspring you would produce. For our genotypes, we have one box out of 4 as big B, so that would be 25% BB. We have 2 out of the 4 boxes being BW, so that'd be 50% heterozygous. And then the other box, 25% would be WW. For phenotypes, the BB would code for black wool, so that's 25% black wool. The 50% heterozygous, that BW, will code for gray, so it would be 50% gray. And then the WW will code for white wool, so that 25% will be white. And that's going to be it for incomplete dominance.